In the waters off the coast of Naples, a common octopus sits motionless in a research tank, missing one of its 8 arms. What if you could lose part of your nervous system, the wiring that controls movement, sensation, even your ability to breathe, and simply grow it back, better than new, in a matter of weeks? The octopus does not panic. It does not seem distressed. Within hours of the injury, something has already begun. Deep inside its tissue, at a level invisible to the naked eye, molecular switches are flipping. Genes are activating. Chemical signals are flooding toward the wound site. The octopus is not healing in the way humans heal, sealing the damage with scar tissue, accepting the loss as permanent. It is preparing to rebuild completely. Arm, nerves, suckers, muscle, skin, all of it, as if the injury never happened. And this is not some rare ability triggered only in extreme circumstances. This is standard operating procedure. For an octopus, regeneration is not extraordinary. It is Tuesday. Octopuses belong to the Cephalopod family, a group that includes squid, cuttlefish, and nautiluses, animals that have fascinated scientists for centuries because of their intelligence, their alien nervous systems, and their uncanny ability to manipulate their own bodies. But even among cephalopods, the octopus stands out. It does not just change color or squeeze through impossibly small spaces. It can lose a limb and reconstruct it from scratch, restoring not just the physical structure but the full functionality, the precise coordination of millions of nerve cells, the sensitivity of hundreds of suckers, and the muscular control needed to manipulate objects with the same dexterity as before. Most animals cannot do this. Humans certainly cannot. If you sever a nerve, it might heal partially, but it will never be the same. If you lose a finger, it is gone. Your body will close the wound, form scar tissue, and move on. The octopus does not accept this limitation. And researchers are finally beginning to understand how. The octopus arm is not a simple appendage. It is more like a semi-independent organism. Each arm contains roughly 2/3 of the octopus's total neurons, around 40 million nerve cells per arm. These neurons are organized into clusters called ganglia, which act as localized control centers. The arm does not need constant input from the brain to function. It can taste, touch, and even make decisions on its own. A severed octopus arm will continue moving for minutes after separation, still exploring its surroundings, still responding to stimuli. This distributed nervous system is one of the reasons octopuses are so cognitively unusual. Their intelligence is not centralized the way ours is. It is spread throughout their body. And when an arm is lost, the octopus does not just lose muscle and skin, it loses a significant portion of its neural capacity. But within 3 days, the regeneration process has already begun. The first visible sign is the formation of a small knob at the injury site. This knob is made of undifferentiated cells, cells that have not yet committed to becoming muscle, nerve, or skin. They are waiting for instructions, and those instructions come from a cascade of molecular signals that scientists are only now beginning to decode. One of the key players in this process is a protein called acetylcholinesterase, spelled ACHE. You have acetylcholinesterase in your body too. It is primarily known for its role in the nervous system, where it breaks down the neurotransmitter acetylcholine at synapses. But acetylcholinesterase also plays a role in cell growth, differentiation, and cell death. In octopuses that are regrowing limbs, acetylcholinesterase activity spikes dramatically, but only at specific times. Researchers studying the common octopus, Octopus vulgaris, discovered this by removing small tips from the arms of 6 healthy females and tracking their recovery over several months. What they found was a precisely timed sequence of events, orchestrated by molecular signals that turned on and off like switches. For the first 2 weeks, acetylcholinesterase activity remained low. The knob formed. Stem cells and blood vessels arrived at the injury site. By week 2, a hook-like structure appeared at the end of the regenerating arm, a kind of scaffold that would guide the growth of new tissue. Then, around day 21, everything changed. Acetylcholinesterase activity surged, and at the exact same time, the first new suckers began to appear. Chromatophores, the color-changing cells in the octopus's skin, started forming. Muscles developed. Nerve fibers extended. The arm wasn't just growing back, it was differentiating. Cells were receiving specific instructions. Become a sucker, become a nerve, become skin. And they were following those instructions with extraordinary precision. By day 42, the acetylcholinesterase activity began to drop. By day 130, the arm was fully regenerated, and acetylcholinesterase levels had returned to normal. The regenerated arm was indistinguishable from the original. Same structure, same function, same neural density. This isn't the sloppy regeneration you see in lizards, where the regrown tail is often shorter or less functional. This is complete reconstruction, and it happens every time. But the octopus's regenerative abilities do not stop at the arms. Scientists at the Stazione Zoologica Anton Dohrn in Naples discovered something even more remarkable. Octopuses can regenerate parts of their central nervous system. Specifically, they can regrow the pallial nerve, a critical nerve that runs from the brain to the mantle, controlling both respiration and the color-changing ability that octopuses use to communicate. The pallial nerve serves 2 essential functions. First, it controls the respiratory muscle in the mantle, which expands and contracts to pump water over the gills. Without this nerve, the octopus cannot breathe properly. Second, it regulates the chromatophores in the mantle skin, allowing the octopus to form complex color patterns. Octopuses use these patterns to communicate with each other, to camouflage, and to express emotional states. Lose the pallial nerve, and you lose both the ability to breathe normally and the ability to communicate. The researchers anesthetized 14 wild octopuses and carefully severed one of the 2 pallial nerves with fine scissors, leaving the other intact as a control. Immediately after the surgery, the effects were dramatic. The respiratory muscle on the affected side of the mantle stopped contracting. The skin turned from its normal brown to a ghostly white because the chromatophores were offline. Then the researchers did something unexpected. They put live crabs in the tanks. The octopuses, still groggy from anesthesia, immediately attacked the crabs. No hesitation, no signs of distress. The damage to the pallial nerve had not affected their hunting ability or their general behavior, but it had severed a critical connection between the brain and a major part of the body. Over the next several weeks, the researchers watched as the nerve slowly repaired itself. Within a few weeks, small brown spots began to appear on the white mantle skin. These spots formed simple color patterns when the octopus was at rest. By 6 weeks, the mantle had returned to its normal coloration, and the octopus was forming full color patterns during hunting and movement. 4 weeks after the surgery, the respiratory muscle began contracting again. The mantle opening resumed its rhythmic expansion and contraction. The octopus was breathing normally, but behavioral recovery was not enough. The scientists needed to confirm that the nerve itself had physically reconnected. 45 days after the injury, they dissected the regenerated tissue and found that the two severed ends of the pallial nerve, one connected to the brain and one to the mantle, had grown back together. The nerve fibers had crossed the injury site and formed a functional network with the local nerve cells in the mantle. To prove that the regenerated nerve was truly functional, the researchers placed electrodes on either side of the lesion. They stimulated the nerve on the brain side and recorded the electrical activity on the mantle side. The signal passed through. The regenerated nerve was conducting electricity just like the original. For the first time, scientists had confirmed that an octopus can completely regenerate a critical component of its central nervous system and restore full functionality. This is not supposed to be possible. In mammals, nerve damage is often permanent. Spinal cord injuries do not heal. Severed nerves may form partial connections, but they rarely restore full function. We have built entire fields of medicine around managing nerve damage because we assume it cannot be fixed. The octopus disagrees. So what is happening at the molecular level that allows this? The answer appears to involve the same mechanisms that drive arm regeneration: stem cells, precise chemical signaling, and the activation of specific genes at specific times. Acetylcholinesterase spikes during arm regeneration, and it is not just involved in breaking down neurotransmitters. It also influences cell proliferation and differentiation. When acetylcholinesterase is active at the right time and in the right place, it seems to guide undifferentiated cells toward becoming the specific types of tissue needed for regeneration. But acetylcholinesterase is not working alone. There are likely dozens, maybe hundreds, of other molecular signals involved. Some are turning genes on, some are turning genes off, some are guiding stem cells to the injury site. All of this is happening in a coordinated sequence that unfolds over weeks and months, following a biological blueprint that the octopus carries in its DNA. The implications for medicine are staggering. If we could understand how the octopus regenerates nerves, we might be able to develop treatments for spinal cord injuries, stroke, or neurodegenerative diseases. If we could replicate the molecular signals that guide octopus tissue regeneration, we might be able to regrow damaged organs or lost limbs. The octopus is not just regrowing tissue. It is rebuilding functional structures with all the complexity and precision of the original. And it is doing this without forming scar tissue, without inflammation spiraling out of control, without the immune system attacking the regenerating tissue as foreign. The process is clean, efficient, and repeatable. Other animals can regenerate too. Zebrafish can regrow heart tissue. The Mexican axolotl salamander can regenerate entire limbs, but most of these animals are distant from us evolutionarily. The octopus, despite being an invertebrate, has a complex nervous system that shares some fundamental features with our own. But there is a deeper mystery here. Octopuses do not live long. Most species live only 1 to 2 years. They do not have extended childhoods. They do not learn from parents. A young octopus, only months old, can lose an arm and regenerate it perfectly on the first try with no prior experience. This suggests the regenerative program is largely genetic, hardwired into the octopus's DNA. But if that is true, why do not all animals have this ability? The answer may be environmental. Octopuses are soft-bodied animals living in a dangerous ocean filled with predators. They rely on speed, camouflage, and intelligence to survive. Losing an arm to a predator is common. An octopus that can regrow that arm has a massive survival advantage. Mammals evolved on land, where injuries of that magnitude are often fatal anyway. Evolution did not select for regeneration because it was not worth the metabolic cost. But the octopus needed it, and so it kept it. Somewhere in the ocean right now, an octopus is regrowing an arm. Stem cells are migrating to the injury site. Chemical signals are orchestrating the formation of new tissue. Genes are turning on and off in a precise sequence, and within weeks, the arm will be whole again. We are watching an animal rebuild its own body, and we are only just beginning to understand how.